Bismillahirrahmanirrahim. Assalamu alaikum. I am here from Conceptual Medicos, and today we are going to talk about the genus Compylobacter. So this genus has two important species. The first one is the Compylobacter jejuni, and the second one is the Compylobacter intestinalis, in which the Compylobacter jejuni is very important. So the common features of this genus is they are gram-negative rods with S shape or comma shape. They are microaerophilic. To understand this is important means they can grow in 5% of oxygen rather than 20% of oxygen, which is the atmospheric percentage. The Compylobacter jejuni grows in 42 degrees centigrade, while the C. intestinalis doesn't grow, and this is one of the differentiating point between the two. Coming toward the transmission, so basically the feces of the cattle, chicken and dogs first contaminate the water and food, and through this contaminated food and water the organism enter into the human and cause the disease in human. The second main source is the unpasteurized milk, poultry, meat, etc. From human to human, the transmission is less as compared to animal to human. The next one is the epidemiology. So in the USA, from about 4.6% of the diarrheal patient, the Clostridium jejuni is recovered. A very high rate from Salmonella, which is 2.3%, and from Shigella, which is 1%. Now coming toward the pathogenesis, so basically this organism attacks the colonic mucosa, and here it colonizes itself and it causes invasive inflammation, unlike to that of the other organism, the toxigenic organisms, which causes bloody diarrhea. Okay, and in neonates it causes bacteremia. Coming towards the clinical findings of that of the Clostridium jejuni are: first, there is watery diarrhea which has foul smell. Then it is followed by bloody stools because it is an invasive inflammation. So there will be bloody stools having neutrophils, and there will be fever and abdominal pain. Okay, and basically the Campylobacter intestinalis causes bacteremia and other systemic infections. Coming towards very important point about the Clostridium jejuni, the Campylobacter jejuni infections, that it is the main cause of the Guillain-Barré syndrome and reactive arthritis or Reiter syndrome. So how it happens that basically the antibodies which are formed against the C. jejuni will attack the antigens on the neuron and will destroy them. So it will cause acute neuromuscular paralysis. About the Reiter syndrome, so in this case the joints, the urinary tract and the eyes are affected, and you can find this by the common triad that the person can't see, can't pee and can't climb the tree. Means the eyes, the joints and the urinary tract are infected. Okay, coming to the lab diagnosis, so stool specimen is taken and cultures on the scarose medium. Scarose medium is basically a blood agar with antibiotics added to inhibit the growth of the normal flora. The antibiotics added are vancomycin, trimethoprim, cephalosporin, etc. Okay, this plate is then incubated in 5% oxygen as the organism is microaerophilic and 10% carbon dioxide. Okay, the organism, the grown organism will be C. jejuni here. Okay, the organism will be failed to grow in 25% oxygen because it will grow at 42 degrees centigrade. 25 degrees centigrade, it will fail to grow at 25 degrees centigrade and will grow at 42 degrees centigrade. The organism is oxidase positive and sensitive to nalidixic acid. Note that only CJ is sensitive to nalidixic acid, and Compylobacter intestinalis is resistant. Treatment is for the C. jejuni is ciprofloxacin and erythromycin, while that of C. intestinalis is aminoglycoside. Thank you.